We now welcome to the stage Jennifer, Anna and Kajal from the Lycée Michel Lucius. They want to reduce plastic pollution by using genetically modified E. coli to code for specific enzymes that break down plastics. Stay tuned for more details on their project, titled Degradation of Plastic Polymers Using Genetically Modified Bacteria.
Good morning, ladies and gentlemen. I'd like to introduce myself. My name is Jennifer Michael, and these are my teammates Anna Batmanova and Kajal Karki. We'd like to present our topic, which is the degradation of plastic polymers using genetically modified bacteria. So why did we choose this topic? We wanted to find a sustainable method to reduce plastic pollution in Luxembourg's rivers and rivers worldwide. Plastic pollution is defined as the accumulation environment of synthetic plastic products to the point where they cause problems in wildlife and its habitats, as well as for human populations.
Plastics are non-biodegradable, meaning it will take hundreds and thousands of years for it to be broken down in nature, which then causes an accumulation of such plastics in landfill sites. So with our project, we want to find a sustainable method with which we can speed up this process of degradation and so decrease the problem of plastic pollution in the environment.
Another reason for doing this project is that during my internship week at the University of Luxembourg, I saw how much plastic was used every day. After that, I discussed this with my friends, and we came up with a few interesting ideas.
So this is our proposed method, which is extremely summarized. And unfortunately, this is all only theoretical, as we couldn't do this practical due to COVID-19 restrictions.
Foreign DNA coding for enzymes, PETase and metase, was put into our vector Escherichia coli, also known as E. coli. E. coli transcribes the gene on foreign DNA that codes for the enzyme.
The bacteria then translates this code to make the amino acids, and then those amino acids come together to make our enzymes.
We then tested how these enzymes break down plastics, specifically focusing on PET.
More in detail, we assume that we will start off with only one strand of DNA. To multiply the DNA to a big enough sample, we use polymerase chain reaction, also known as PCR. To modify the bacteria, we use the restriction enzyme to cut the part of the plasmid. DNA ligase was used to join the sugar phosphate backbone of DNA coding for the metase and petase. And finally, the double-stranded DNA was produced.
The next step was to insert the genetically modified plasmid into the bacteria. This was done by adding concentrated calcium ion solution into the Petri dishes containing the bacterial culture. The bacterial culture was then given a heat shock, which made sure that the plasmid passed through the cell surface membrane. The initial DNA coding for petase and metase was given a genetic marker, protein GFP originating from jellyfish. The GFP protein fluorescence if it was exposed under UV light. So this can be used to see if the plasmid has been taken up. E. coli is expected to fluorescent if it has been taken up by the modified plasmid.
The next part of our method was to replicate the same bacteria that have taken up the modified plasmid. We achieved this by separating the luminescent bacteria from the non-luminescent ones, and then allowing the luminescent ones to reproduce by binary fission. This ensured that many copies of the same modified plasmid was produced, and then so many copies of our enzymes could be also produced. And then we would introduce our PET plastic into this luminescent bacterial culture for it to degrade the plastic, and we would then record the change in mass.
So this is our theoretical results. We stated that PET alone is going to break down most of the plastic compared to metase-only modified bacteria, which is going to break down the least amount of plastic. The combined enzyme petase and metase is going to break the most amount of plastic in its given time because the enzymes work in a two-enzyme system to break down the plastic.
So as with every experiment, there's also going to be some advantages and, however, also some inconveniences. So from the advantages, we aim to use microorganisms to reduce the problem of plastic pollution, as well as save energy and achieve to have cleaner oceans. So today, most of the plastic that has been disposed of is incinerated. which then causes a loss of materials as well as energy. So with the use of microorganisms, we can prevent this from happening and still have this useful energy and materials for other fields.
The disadvantage of our experiment is that our experiment apply only to PET plastics, as the enzymes are very specific, and we also assume that the process is going to be slow, as the turnover rate of the enzyme is very low.
So looking into the future, we want to investigate our experiment, actually do our experiment, in order to make further discoveries and help solve the problem of plastic pollution. We also want to find different methods for different types of plastic other than PET, as mentioned before.
As mentioned before, the enzymes already have a slow turnover rate. So in the future, we also want to increase this turnover rate of our enzymes PETase and MHETase, so that the degradation of plastics will be faster, and so this method of degradation will be more efficient.
And finally, we want to apply our experiment here in Luxembourg and measure the plastic content in the Luxembourgish rivers and the lakes. Thank you.
Thank you for listening.